Welcome to video on purines and their importance in biological systems. Purines are organic compounds that play a crucial role in the human body, particularly in processes such as DNA and RNA synthesis. In this video we will explore the different pathways of purine synthesis, their regulation, and the various disorders and medications related to purine metabolism. There are two types of purines, adenine and guanine. These molecules are synthesized within cells through complex biochemical pathways, and their levels are tightly regulated to ensure proper cellular function. While purines are important for many aspects of cellular metabolism, excessive accumulation of these compounds can lead to a variety of health problems. Purines are nitrogen-containing compounds that play a vital role in the human body. They are involved in many crucial biological processes, including DNA and RNA synthesis. In fact, purines are the building blocks of DNA and RNA, which are responsible for carrying genetic information in all living organisms. Without purines, these processes would not be possible, and life as we know it would not exist. Purine synthesis is a crucial process in the human body, and there are two pathways involved in this process, the de novo pathway and the salvage pathway. While both pathways are important, they have some key differences. The de novo pathway involves the synthesis of purines from scratch, using amino acids, CO2, and energy in the form of ATP. On the other hand, the salvage pathway involves the recycling of purines from nucleic acid breakdown, which conserves energy. The salvage pathway is also responsible for maintaining adequate levels of purines in the body, as it can synthesize purines more efficiently than the de novo pathway. It begins with the conversion of ribose 5-phosphate to phosphoribosylpyrophosphate, PRPP, by the enzyme PRPP synthetase. PRPP is then converted to 5-phosphoribosylamine by the enzyme glutamine phosphoribosyl aminotransferase, GPAT. The next step in the pathway involves the addition of a second nitrogen atom to form the purine ring system. This is accomplished through a series of reactions catalyzed by the enzymes phosphoribosylglycinamide synthetase, phosphoribosylglycinamide transformylase, and phosphoribosylaminoamidazole synthetase. The final product of this pathway is inosine monophosphate, IMP, which can be further converted to adenosine and guanosine nucleotides. The key enzymes involved in the salvage pathway include hypoxanthine guanine phosphoribosyltransferase, HGPRT, and adenine phosphoribosyltransferase, APRT. These enzymes catalyze the conversion of hypoxanthine and guanine, or adenine, respectively, into their corresponding nucleotides, which can be used in DNA and RNA synthesis. The salvage pathway is particularly important in tissues with high turnover rates, such as the liver and bone marrow, where large amounts of purines are constantly being degraded and recycled. One of the key mechanisms involved is feedback inhibition, where the end products of purine synthesis act as inhibitors of the enzymes involved in their own synthesis. This helps to prevent excess purine production and maintain homeostasis. Another important mechanism of regulation is enzyme activity modulation, where the activity of the enzymes involved in purine synthesis is controlled by various factors such as allosteric regulation and post-translational modifications. These mechanisms allow for precise control over purine synthesis, ensuring that it occurs only when needed and in the appropriate amounts. Gout is a type of arthritis that occurs when uric acid crystals accumulate in the joints, leading to inflammation and pain. Lesch-Nyhan syndrome is a rare genetic disorder that affects the nervous system and causes severe mental and physical disabilities. Purine nucleoside phosphorylase deficiency is a rare inherited disorder that affects the immune system and can lead to recurrent infections. Lesch-Nyhan syndrome is a rare genetic disorder that affects the metabolism of purines, resulting in the overproduction of uric acid. This condition is caused by mutations in the HPRT1 gene, which encodes for an enzyme involved in purine metabolism. Symptoms of Lesch-Nyhan syndrome include neurological and behavioral abnormalities, such as self-injurious behavior, gout, and kidney stones. Currently, there is no cure for Lesch-Nyhan syndrome, and treatment options are limited to managing the symptoms of the condition. Purine analogs are synthetic compounds that mimic the structure of natural purines. They are used in medicine to treat a variety of conditions, including cancer and autoimmune diseases. One example of a purine analog is azathioprine, which is used to suppress the immune system and prevent rejection in organ transplant patients. Another example is mercaptopurine, which is used to treat leukemia and other cancers by interfering with DNA synthesis. One example of a purine synthesis inhibitor is mycophenolate mofetil, which is used to prevent organ rejection in transplant patients. This drug works by inhibiting the enzyme inosine monophosphate dehydrogenase, which is involved in the de novo pathway of purine synthesis. By reducing the production of purines, mycophenolate mofetil helps to suppress the immune system and prevent rejection of the transplanted organ. Mycophenolate mofetil is a medication that is used to prevent rejection in patients who have received an organ transplant. It works by inhibiting the production of purines, which are necessary for the replication of T and B lymphocytes. This leads to a decrease in immune cell proliferation and ultimately prevents rejection. Allopurinol is a medication used to treat conditions caused by excess uric acid in the body, such as gout and kidney stones. It works by inhibiting the enzyme xanthine oxidase, which is responsible for the production of uric acid. By blocking this enzyme, allopurinol reduces the amount of uric acid produced in the body, helping to prevent the formation of uric acid crystals that can cause painful inflammation and damage to joints and other tissues. Like, share and subscribe for more such videos.